Everyone. Today I'll be talking about a very detailed survey that we did here at HealthTrackRx about the bacterial and viral co-infections of SARS-CoV-2. And to our knowledge, this is the largest study or largest such study into the co-infection rates of this novel coronavirus. Disclosure slide here. So the SARS-CoV-2, which is a latest coronavirus member, the coronavirus family named as such due to the characteristic distribution of the spike protein on the viral surface, is responsible for causing from mild to fatal respiratory illnesses in humans. And when we compare the epidemiology of this viral disease with the other known respiratory pathogens, we find the infectivity of this virus is somewhere between influenza and SARS-CoV-1, which was responsible for the 2002 SARS pandemic. And since the first official declaration of this viral outbreak at the end of 2019, the global pandemic has infected over 29 million people. The numbers that I have here are out of date now. And within the United States, there are over 6.5 million confirmed cases of the viral infection and more than 194,000 deaths. And what we have seen in recent memory, the most unprecedented mobilization of public health resources in our fight against this virus, which has had a great impact on our economic and social behavior. And quite literally, our knowledge of how this virus works and the COVID-19 disease that it causes, our knowledge of this virus is increasing every day. However, what we find is that the co-infections or the bacterial and viral co-infections of SARS-CoV-2, that's an area where our understanding is still quite limited. Now, what we know from our experience with the other respiratory viruses is that the concurrent or secondary co-infections of these viruses with other bacteria lead to an increased morbidity and mortality in patients. And in fact, the general consensus now is that all the influenza pandemics of the last century, the leading cause of mortality in those patients were the secondary or co-infections with bacterial pathogens. And this is due to the fact that these respiratory pathogens, both the bacteria and the viruses, have co-evolved in a manner to take advantage of the preceding infection. So here at HealthTrackRx, co-infections of respiratory pathogens is an area that we're always interested in. And in 2019, we had published a very detailed analysis of co-infections in respiratory viral pathogens, influenza, RSV, and others. And the present data set, like I said, is the largest study that we know of. It comprised of 50,419 individual samples that were tested between mid-March and the beginning of August this year. And the samples were concurrently tested for SARS-CoV-2 using the TACPath COVID-19 kit from Thermo Fisher, and all the other pathogens, both bacterial and viral respiratory pathogens, were tested on the nanofluidic open array platform, also from Thermo Fisher. And what we found was that the SARS-CoV-2 infection rate at 8.4% was very similar to all the other viral pathogens put together at 8.2%. We found in the overall population a bacterial infection rate of around 33%. Now, When we do a demographic distribution of the SARS-CoV-2 infections based on gender, we find that the female and male ratio was quite similar to the overall population set that we had, with nearly 56% females and 44% males. And when we distribute the same according to age, what we found was quite interesting is that overall, the younger population, that is between 20 to 50 years of age, comprised, both male and female, comprised a higher proportion as opposed to the older population, that is 55 or 60 years and higher. When we look at the types of co-infections, what we find is that the distribution of the bacterial co-infections in both the SARS-CoV-2 positive and SARS-CoV-2 negative population is quite similar. The numbers are, statistically speaking, are significantly lower in the SARS-CoV-2 positive population, but, still comparable at 33 and 35%. However, this difference is more pronounced in the viral co-infections, where in the SARS-CoV-2 positive population, the viral co-infection rates are much lower as compared to the SARS-CoV-2 negative population. And when we actually go into the different types of co-infections, with the bacterial co-infections, we find the usual bacterial pathogens, namely Streptococcus pneumoniae, Staph aureus, Haemophilus influenza, that are represented as the major co-infecting agents. Klebsiella pneumoniae also, but to a lower level as compared to these other pathogens. But what we see is that the bacterial infection rates or co-infection rates in the SARS-CoV-2 positive population are lower as opposed to the SARS-CoV-2 negative population, with the exception of Staph aureus, wherein the infection rates are statistically significant and higher in the SARS-CoV-2 positive. Now, with the informal discussions with other labs, we find that this is not a unique observation at our end, and the other labs are also seeing this. And we find this quite interesting, and this is something that we are looking at much more closely now, is to basically figure out what percentage of this is represented by the methicillin-resistant Staph aureus or MRSA, because that has a very significant impact on the morbidity and mortality that is associated with respiratory infections. Now, we also found in the general population, we found less than 0.1% infection rate of Chlamydophila pneumoniae, Mycoplasma pneumoniae, and Bordetella pertussis, and no co-infections with those pathogens, and that is why that is not represented here. When we look at the viral co-infections, the majority of the pathogens, as you will see here, represent a less than 1% prevalence rate. And that could very well be due to the fact that when we started collecting this data, it was towards the end of March. And that coincides with the end of the respiratory pathogen season, or the flu season here in the United States. And again, what we see here with the blue bars, the SARS-CoV-2 positive population, the co-infection rates of these viruses are lower as opposed to the SARS-CoV-2 negative population, with the exception of RSV. And again, I apologize for some confusion here. The HSV here is a combination of HSV1 and 2, and the HSV at the end here is HSV3, or the varicella zoster virus. And these are the only two viruses wherein we see a higher co-infection in the SARS-CoV-2 positive population. Now, the other exception that we saw was EBV, the Epstein-Barr virus, or HSV4, and the rhinoviruses, where we see a higher than 1% prevalence rate in the general population, and a significantly higher EBV co-infection rate was also seen in the SARS-CoV-2 population. And if you go through the latest literature, what you find is that these EBV co-infection or higher EBV co-infection rates in SARS-CoV-2 positive patients have been reported from other sources also. When we look at the age distribution of these co-infections put together, both bacterial and viral, in the SARS-CoV-2 positive population, what we find is that the older population, 60 plus years, they are more susceptible to these co-infections. And again, if you would recall, the age distribution of the SARS-CoV-2 positive population was more towards the younger side. However, the co-infections are higher in the older population. And this could very well contribute to the poor prognosis of the COVID-19 disease that we observe in the older population. And again, what we are doing right now, since, as I mentioned before, most of this study was done towards the end of the flu season. So what we are doing right now is that we are actively collecting data now that fall is upon us and winter is coming, quite literally, that we will be looking at these co-infections in much more detail, and we'll be presenting a follow-up of the study towards the end of the flu season in 2021. So to summarize, what we see is that the SARS-CoV-2 population, positive population, displays a significant co-infection of both bacterial and viral pathogens. And generally speaking, the viral co-infections in the SARS-CoV-2 positive population were found to be significantly lower as opposed to the SARS-CoV-2 negative population. And within the viral co-infections, we find that the EBV or the Epstein-Barr virus co-infections are detected at a significantly higher rate in the SARS-CoV-2 positive population when compared to the all other respiratory viral pathogens. And amongst the bacterial pathogens in the SARS-CoV-2 positive population, we find a significantly higher co-infection rate of Staph aureus. And when distributed against age, what we find is that put together, the viral and bacterial co-infections are observed at a higher rate in the older population, that is 60 years of age and higher, as opposed to the all other age groups. And what we believe is that the data presented here in the follow-up study that we are doing right now will allow for a better clinical decision to be made with respect to the antimicrobial therapy that can be provided to the infected subjects for for better clinical treatment. And with that, I would like to acknowledge the infectious disease team here at HealthTrackRx, namely my co-authors on this study, Dr. Jay Reddy, the VP of Lab Operations, and Dr. John Granger, our medical director, and all the other scientists and laboratory personnel working here at infectious disease in HealthTrackRx. And with that, I'll take any of your questions. Thank you.